And a very warm welcome to each and every one of you. Thank you so much for joining us for today's webinar. This webinar is hosted by Jen and sponsored by Asimov. Today's topic is next-gen lentiviral vector manufacturing, improving scalability, control, and quality. Thank you so much for joining us. I'm Uduak Thomas, Senior Editor at Jen, and I'm delighted to be your host for today's webinar. First things first, I'd like to introduce today's speaker. You'll be hearing from Dr. Brianna Jayanthi. She's a principal scientist at Asimov. We'll get to her presentation shortly, but first, a little bit of background on today's topic. Lentiviral vectors, or LVVs, are widely used as a delivery system for ex vivo cell and gene therapies. Now, in recent years, there is growing interest in using them for in vivo therapies, and that is driving new demand for high quality LVVs at scale. Transient transfection systems, which are the dominant method used for LVV production, have well documented limitations in scalability and product quality. So, in response, therapeutic developers have begun developing stable LV production systems, which promise to bring LVV manufacturing in line with gold standard practices used for protein biologics production. So, in this webinar, you'll hear from our speaker how Asimov's LV Edge producer and packaging systems lead to higher quality, higher productivity cell lines for clinical and commercial LV therapies. You'll hear about a case study evaluating transient, semi-stable, and stable LVV production systems through a manufacturing and product quality lens. And you'll also get some guidance to help you determine which production system is best suited for various therapeutic cases. Now, today's presentation is going to be followed by a live Q&A. So at any point during today's webinar, if you have a question, feel free to enter it in the Ask a Question panel on your screen, and we'll do our best to get to your question before the top of the hour. And with that, I'm going to hand things over to our speaker for her presentation. Brianna, the floor is yours.
Thank you, Udak, and thank you all for joining today. It's my pleasure to tell you about Asimov's LV Edge system in this webinar, Next-Gen Lentiviral Vector Manufacturing, Improving Scalability, Control, and Quality. I'm planning to take you through a brief overview of Asimov and our LV Edge system. Then I'll share with you two case studies. One where we compare the production of lentiviral vectors in LV Edge packaging and producer systems with an off-the-shelf 4-plasmid transient system. And another case study where we worked with a CDMO to compare critical quality attributes of product made in the LV Edge packaging or producer systems. Finally, I'll wrap up with a brief recommendation of how these two production systems, the semi-stable packaging and the fully stable producer, work together to meet production needs. First, let's talk a bit about Asimov. So Asimov is a tools company with a unique integration of software with biology. We develop and GMP bank host cell lines. We mine and optimize genetic parts and systems, and we build data-driven models and combine these into our own software for computer-aided design of biological systems. This ecosystem is easing the design and manufacture of advanced therapies. We have two models for how we engage with partners, one where they in-license our technologies and use them at their own site, and the other where we do the requested work for cell line development, process development, and analytics here at our Boston, Massachusetts headquarters. We currently have 51 biopharma partners that we have worked with across 69 drug programs. And our modeling approach is structured along two dimensions, our workflows and a mechanistic statistical nature. We have platforms that span the advanced therapy space, from CHO-based biologics production, where we excel at generating high-titer cell lines for difficult-to-express molecules, to the more nascent area of RNA therapies, where we have unique capabilities for tissue-specific expression, as well as proven product stability with an in vitro transcription system. In the cell and gene therapy space, for AAV production, Asimov has both a 2-plasmid transient and a fully stable producer system that we compare with our validated tissue-specific promoters. Today, I'm going to focus on our systems for lentiviral vector production, where we use a HEC293 host cell line free of the SV40T antigen to enhance the safety of our lentiviral vector product. Like other advanced therapies, the current typical paradigm for lentiviral vector manufacturing has unsolved challenges related to cost and risk, scalability, and reliable product quality. Thinking about cost and risk, the expensive multiplasmid transient workflows, high batch failure rates due to complex and often still manual transfection operations, the supply chain constraints for GMP plasmids, as well as other supplies like transfection reagent, can all lead to an increased risk of timeline changes that disrupt manufacturing and clinical plans. When it comes to scalability, low volumetric productivity limits the feasible batch yield. Complex transfection processes also challenge scale-up and tech transfer, and a need to scale out manufacturing often means large facility footprints can be required to meet those vector needs. With current production methods, it's often not viable to meet the demand for indications with large patient populations. The transient production process that are typical for lentiviral vector or LVV manufacturing often have a high proportion of product and process impurities, high batch variability when it comes to potency and infectivity, and these processes can be really difficult to control CQAs and impurities due to their transient nature. These hurdles are further compounded by limited analytics or non-standard analytics to characterize vector product heterogeneity. Asimov's LVH system is really shifting the paradigm for lentiviral vector manufacturing, away from transient transfection with stable cell lines to mirror the more familiar, scalable, and high productivity manufacturing processes of biologics. I'll just walk you through what cell line development and manufacturing looks like for a typical transient system and for Asimov's semi-stable packaging and fully stable producer systems. Transient processes do not require any cell line development ahead of manufacturing, and the production is typically carried out by transient transfection of 4 plasmids: GAGPOL, REV, pseudotype, typically VSVG, and a gene of interest or transfer vector. The LVH packaging system is an off-the-shelf system, meaning the cell line development piece is already done. We stably integrated the lentivirus packaging components, GAGPOL, REV, and VSVG pseudotype, and selected a clonal cell line. This clonal cell line is then used to manufacture lentivirus by transiently transfecting a single transfer plasmid containing the gene of interest, and expression of the viral packaging components is induced with addition of the small molecule doxycycline. The LVH producer system requires a short 4-month custom cell line development. The producer system is fully stable, meaning we integrate the lentivirus packaging components, GAGPOL, REV, and a pseudotype of choice, as well as the transfer vector containing a gene of interest. We go through screening and selection of a high-producing clonal cell line. This clonal cell line is then used to manufacture lentivirus by simply inducing the expression with addition of doxycycline. When we look at the trade-offs between these systems across cost of goods, process complexity, reproducibility, and the ability to tune product quality attributes, we see a similar trend where the transient system has high risk or a more challenging process, and stable systems reduce that risk and ease manufacturing. Focusing on cost of goods, the transient processes have high cost of goods by requiring this 4 plasmid and transfection reagent, all GMP grade, variable productivity, and unscalable transfection processes also impact the cost of goods. The packaging system lowers cost of goods by reducing the number of plasmids required to transfect, but the transfection process is still variable and can limit scalability. The producer system has the lowest COGS with a system free of the need to transfect and higher productivity and scalability. When it comes to process complexity, the transient processes are more complex due to the transfection, which still often requires manual operation. The packaging system reduces the process complexity by limiting the number of plasmids from 4 down to 1. This can also make process optimization easier. The producer system has the most streamlined operations with no transient transfection and only needing the addition of small molecule doxycycline to induce production. When it comes to reproducibility, the inherent variability of a 4-plasmid transient process leads to increased risk of batch failures and product quality variations. The packaging system decreases that risk by limiting the transfection step to that single plasmid. The producer system has the most reproducible production processes because the variability due to transfection is removed and gene ratios in producer cells are consistent. For product quality attribute tunability, the transient production system is limited because the production processes are typically dependent on optimizing the transfection rather than growth and feed strategies. The packaging system has the same transfection constraint on optimizing the production process, while the producer system has flexibility to tune product profiles through clone selection and also process flexibility by removing that transient transfection step. Now that I've introduced you to the challenges we aim to solve with the LVH system, let me share with you some of the data. At a high level, the semi-stable packaging system is a cell line ready to transfect with your transfer or GOI plasmid, and production is initiated with addition of doxycycline. This system best supports rapid timelines and with optimized DNA design and platform processes. Using this system, we routinely achieve mid-E8 functional titers across anti-CD19 and anti-BCMA CAR payloads. And productivity is improved with our bioreactor processes. In this chart, we are comparing crude harvest lentiviral vector in a transduction assay. We transduced the permissive HEC293T cell line and measured integrated copy number by ddPCR. In the AMBER 250 system, we achieve between 5 and 8 times 10 to the 8 transducing units per milliliter. Our fully stable DOX-inducible producer system is best when objectives are to maximize titer, scalability, and reproducibility. Using this system, we achieve up to 10 to the 9 functional titers with crude harvest. A custom clonal producer cell line can be ready in just 4 months. We simultaneously select for integration of the lentiviral packaging components, a pseudotype of choice, and a gene of interest in a fully suspension animal component-free process. We then single-cell print and screen clones with a model-guided approach to progressively advance clones through the workflow. We confirm top clone titers in an AMBER 15 microbioreactor process and then release RCBs to customers. In this chart, I'm showing crude harvest from the AMBER 15 scale measured in our standard HEC293T transduction assay. And here, the top 4 clones across 3 development programs, we see top clones achieve 7E8 up to 1.69 titer per milliliter. Also notable are the high infectivity ratios observed in the unpurified product from top clones, reaching up to more than 900 TU per picogram of P24 protein. With that overview, I now want to jump into our 2 case studies. First, comparing productivity in our packaging and producer systems with commonly used off-the-shelf system for transient production. Then I'll tell you more about an investigation of translatability of the lentiviral vector product from our packaging and producer systems. Our motivation for case study 1 was to compare performance of these different types of production systems in terms of functional titer and batch variability. For this comparison, we looked at one anti-CD19 CAR molecule. Using our internal vector design, we built a transfer vector for this molecule that was used in both the transient and packaging systems. The transient system, again, was off the shelf, and we followed manufacturers' recommended protocols for production. We took the same anti-CD19 CAR molecule through our CLD process to generate a clonal cell line. These 3 production systems were then compared in a 2-day shake flask production. We had 3 production batches starting from vial thaw, each with 3 technical replicates, and all of the measurements were done with at least duplicates. To understand the manufacturability with each system, we looked at cell density and viability at harvest, as well as major process impurity. And we measured both physical particle titer and functional titer in our HEC-293T transduction assay. Our main finding was the LV-Edge packaging and producer systems deliver improved manufacturability and productivity. Looking first at how healthy our cell factories are, we see distinct differences in both harvest cell densities and viabilities between the transient system and LV-Edge systems. The packaging and producer systems average 2.4 to 3 times greater harvest cell densities compared to the transient system. I'm showing the cell densities here on a linear scale, but packaging and producer average 7.5 to 9.5 million cells per mil at harvest, while the transient system is less than half that, averaging around 3 million cells per milliliter at harvest. The packaging and producer systems both maintain high, greater than 90% harvest viability. We also looked at a major process impurity, host cell protein. Perhaps intuitively, with the lower viability, we see a much higher amount of host cell protein in the crude harvest from the transient system, which is reduced 2.8 to 5-fold in the LV-Edge systems. We next compared productivity in a transduction assay, measuring copies integrated into a HEC-293T cell line. We see functional titer increase 3 to 5-fold in the LV-Edge systems, and notably, the variation, which is across batches, is reduced. Looking at the coefficient of variation, which is reduced more than 2x with the packaging system and roughly 4x with the producer system. We also see more than 3x improvement in the infectivity ratio of the lentiviral vector product, moving into semi-stable and fully stable systems, measured as transducing units per microgram of P24 protein. This ratio increases 30% in the semi-stable system and roughly 3.2-fold in our fully stable system. Comparing packaging versus transient systems, we see similar productivity, a more than 2x reduced variability, and 30% improved infectivity moving to the packaging system. Comparing producer versus transient systems, we see a 5x increase in productivity, a 4x reduction in batch variability, and a 3x improved infectivity ratio. Our key findings from this comparison between the 3 production systems are that manufacturability is improved by better cell health through production and harvest. This improvement might also lend itself well to our perfusion or extended duration production. The cost of goods is lowered with the semi and fully stable systems by limiting transfection to a single plasmid or by significantly increasing productivity per batch. The LV-Edge packaging and producer systems also have improved reproducibility of production batches, which lowers risks to manufacturers. Now let's take a closer look at comparing the LVV product from the packaging and producer systems. Our motivation here was to understand the comparability between packaging and producer LVV samples, and we worked with a leading CDMO on a deep analytical analysis. We wanted to understand this comparability in order to know the feasibility to move between these two systems to meet the changing needs throughout the therapeutic life cycle stages. We produced anti-CD19 CAR lentiviral product in our lab. We shipped these samples to an industry-leading lentiviral CDMO, and they carried out purification by sterile filtration and ion exchange. A full analytical panel was completed looking at functional and physical titer, residual DNA, protein composition, and T-cell transduction. Our key finding from this study was that the lentiviral vector product from both the packaging and producer systems is highly comparable in composition and biological effect. Functional titer was measured on crude, filtered, and ion exchanged material. The product from packaging and producer systems retains functional titer across these steps. qPCR integration titers were within 1 to 1.5 fold of the flow functional titer values, and producer sample titers average around 3-fold greater than packaging sample titers. RNA copy numbers are roughly 3.5-fold higher in producer compared to packaging. Here, again, we're measuring the RNA copy number in both packaging and producer samples across purification steps, sterile filtration, and ion exchange. And these RNA copy numbers are consistent with transduction titers, albeit a few-fold higher. The P24 values are comparable across sterile filtered and IAX samples for both LVH systems. Residual host cell DNA was measured by 18SS qPCR for the packaging system, crude and post-purification IAX levels are similar. For the producer system, samples show higher host cell DNA post-IAX, indicating additional polishing steps may be needed. Residual total DNA levels remain stable between crude and post-IAX material, consistent behavior in both systems. Next was mass spectrometry analysis on the crude and purified samples from both packaging and producer. These samples were processed through protein extraction and digestion, then compared in a data-independent acquisition-based mass spectrometry method. More than 6,400 proteins were identified in all samples, showing a high degree of coverage. And we additionally see a high degree of overlap, 85 to 93%, between all cross-comparison samples. Crude and purified packaging and producer samples have 90% or greater similarity in sample composition. And comparing crude and IEX purified samples, we see a slight reduction in overlap, which is consistent with clearance of some host cell proteins through the purification process. Not depicted here, the major lentiviral vector proteins, GAG and the VSVG pseudotype, are the two most abundant proteins in the purified packaging and producer samples. VSVG represents 30% or more of the sample, and GAG represents 6.5 to 8% of the total sample. Here we move to the endpoint, or perhaps the most important analysis, which is how do the lentiviral vector products perform in T-cell transduction or primary cell transduction. Here we see efficiencies improved roughly 3-fold with purification, and the LVV product from both the packaging and producer systems transduce similarly across two independent donors. We see equivalent biological potency of the LVH system, both pre-purification and that same consistency between LVH systems post-purification. Both LVH systems deliver high-quality, clinically relevant vector that's suitable for downstream purification platforms. Our key findings from this comparability study are that functional and physical titers maintained through purification and residual profiles are consistent between samples. LVV proteins are abundant in LVH samples and are enriched with IEX purification. Purified lentiviral vectors from the LVH system efficiently transduce T cells and are comparable between packaging and producer product. Finally, I'd like to discuss some recommendations for how the product from the LV packaging system and the LVH producer system can be used to meet lentiviral vector demands. The packaging and producer systems are complementary but serve different market needs. We can think about this across two axes. First, you can think about a yearly transducing unit demand, which is roughly equivalent to an indication size, a dose size, and clinical progression. You can also think about this as the amount of drug product COGS dependent on the lentiviral vector composition. So a low composition is likely required in settings such as for autologous CAR T therapies. Medium or higher amount would be required for allogeneic. And then moving into in vivo lenti applications, you're likely to need much higher product demands. Can also think about this as with low demand, speed and flexibility might drive the decision making on platform choice, whereas an increasing need for scalability, productivity and reproducibility may be larger drivers in manufacturing platform choice. We see the LV-Edge packaging system as meeting these demands in low or medium need situations, whereas the LV-Edge producer system is exquisitely suited for high demand applications. Our highly productive LV-Edge cell lines enable reliable lentiviral vector manufacturing with simplified operations and a lower risk of production failures. Our off-the-shelf packaging cell line, ready to transfect with your GOI, improves batch reproducibility, and our producer cell lines, while requiring a short cell line development time, can improve productivity and further reduce batch variability. Both systems are suitable for downstream primary cell line transduction. Lowering the COGS through high-yield scalable manufacturing reduces the cost per dose. Our streamlined operations, removing GMP plasmid needs, simplifying production, and de-risking the supply chain. Improved precision from clinical stable step lines that enhance production control and reduce batch variability, and CQA-driven clone selection through genetic and process levers that enable targeted product profiles are possible with the LV-Edge packaging and producer systems. Thank you.
Wonderful, wonderful. Thank you so much, Brianna, for that presentation. We really appreciate you. And to our audience, thank you so much once again for being here. We're going to begin the Q&A session in just a few moments. So if you have a question, now is the time to go ahead and submit that question if you haven't done so already. Now, before we start the Q&A, I did want to let you know that this webinar is going to be available to view on demand in the next 24 to 48 hours. You should get a link with the email that you used to register for this event. So we invite you to share that link with your colleagues who might not have been able to tune in today. And with that, I'm going to go ahead and get started with the questions. The first question that I have for you, Brianna, and once again, thank you so much for that presentation. First question I have for you, have you tested the stability of the packaging and producer systems?
Yes, we have tested stability of our cell lines out to 10 weeks, roughly 70 generations of continuous passaging without selective pressure. And we see excellent productive and genomic stability.
Excellent, excellent. Can you discuss any experience with RCL in your systems? Is the next question.
Yes, we have tested both of our systems for replication competent lentivirus, and we have no positive results. So our cell lines are free of that replication competent virus.
Excellent, excellent. Next question. Have you tested this process with customers?
We have a robust tech transfer process that's been successful across 9 independent customers so far.
Excellent, excellent. And to our audience, thank you so much for sending in questions. As a reminder, if you do have a question, please feel free to enter it in the Ask a Question panel that is located on your screen. Next question, how do you handle tech transfer?
We have a wonderful tech transfer team and we really take a white gloved approach to this, trying to make sure that we're meeting the customer's needs.
Excellent, excellent. Next question I have, are multiple antibiotics used for selection for the producer cell line?
Yes, we do have a multi-antibiotic selection system.
Excellent, excellent. And then the next question I have you looked at any enhancers that have increased yields? Excuse me.
We have. We do see, consistent with published literature, increased production with the PKC agonists.
Excellent, excellent. Next question I have for you. Can you discuss how you handle clone selection and characterization for the producer system?
Yes, we use a high throughput screening approach guided by our models. Unfortunately, much of the specifics are proprietary. I apologize for that.
No worries. All right. Next question I have. What criteria do you use to identify the top clones?
Again, I think that's a similar question to our clone screening approach.
Excellent. Excellent. And the next question I have, you mentioned Asimov uses data-driven models. What types of models are used and how?
Yeah, so we use both mechanistic models and based on first principles and machine learning or statistical based models. We really work to combine these together, which makes our models much more predictive and successful in the lab.
Excellent, excellent. Brianna, thank you so much once again for your presentation and for taking time to answer a few questions for us. We really appreciate you. And to our audience, thank you once again for being here. One last reminder before we go, this webinar is going to be available on demand in the next 24 to 48 hours. Oh, I see one more question coming in. Actually, Brianna, do you mind if we take this one?
Not at all.
All right. Can you go through some, I think maybe, let me edit this a little bit. Can you shed some light on selecting vectors for mutation correction in gene therapies?
I think on how to select the vectors for mutation correction. I think maybe I don't quite understand the question. I apologize.
No worries. That may be one that we can if the speaker if the audience member could rephrase the question, that would be great. I do have a two-part question here for you as well. I might have missed this, but how's the envelope integrity assessed as part of CLD? And second question, how stable is expression across passages? And what genomic integration strategies reduce silencing in long-term cultures? So quite a few questions there.
Yeah, so I think for question one, the Envelope integrity is really assessed at the end of CLD through functional assays. And then stable expression across passages, what genomic integration strategies are used to reduce silencing in long-term cultures. We do use a transposase-based integration method, and that's based on our proprietary Asimov's own transposase system.
Excellent. And I have another question here for you as well. Are multiple antibiotics used for selecting the producer cell line?
Yes, we do have a multiple antibiotic selection approach.
Excellent. Excellent. All right, wonderful. Thank you once again, Brianna. And as I was saying to our audience, you should get a link related to this webinar within the next 24 to 48 hours that will allow you to view the entire webinar on demand from beginning through to the Q&A section of the webinar. So in addition to watching it, once again, please do share the link with your colleagues who might not have been able to join us today. Many thanks again, Brianna, for being here today and for your presentation. I'd also like to thank today's sponsor, Asimov, for sponsoring the event. And I'd also like to thank the superb multimedia group at Sage for all of their support. Thank you guys so much. And most of all, thank you for watching today. For everyone at Gen, I'm Udrak Thomas. Thank you and goodbye for now.